Diez Benavente and is an assistant professor at UMC Utrecht in the Netherlands. And we're super excited to have him here today to present his work recently published on atherosclerotic plaque epigenetic age acceleration predicts for diagnosis and is associated with endothelial-to-mesenchymal transition in humans. So, Ernest, whenever you're ready, go ahead.
So you can see my presentation?
Yes, perfect.
Okay. So yeah, I just wanted to mention before I start digging into it that this work actually was done in collaboration with another postdoc in the lab who since before we actually got this finally published, he actually moved on to industry, but he did a quite sizable amount of the work. So yeah, that was Robin Hartman in our lab. But yeah, to sort of start the story a bit and give you the background, I think probably you all know that atherosclerosis is a disease that progresses during aging. So whether you look at the presence of carotid plaque, the amount of stenosis, or the intima media thickness, which is a clinical measure of degree of atherosclerosis in the carotids. This is actually this progresses over age, and if you look at men and women separately, you can see that there's kind of like a slight delay in females where this happens roughly about 10 years later in women than in men. But in the recent years, there's been a lot of buzz in different fields of medicine actually about the concept of biological age. So rather than looking at someone's chronological age, people think about biological age as this phenomenon where people who might be the same chronological age might have actually aged faster or slower than what you would expect for a given chronological age. And this can be looked at different levels. So whether it's at the tissue level, whether it's at the physiological level, whether it's at the organ level. But yeah, the concept is that potentially looking at some form of biological age might actually be a more proximal measure of health, if you like, than looking at chronological age. And over the last few years, actually, there's been many forms of measures that have been created to measure this biological age. So some of the most famous ones, I think, rely on methylation. Another one that is also quite widely used is telomere length. But over the last 10 years or so, there's been a lot of work looking at different substrates to measure this biological age, including some very novel techniques using, for example, ECGs or MRIs or proteomics. But what I'm going to focus on today mostly is biological age measured as through the use of methylation. For this, actually, we used in this study the Horvath clock. So the Horvath clock is basically if you take there's 353 CPGs within everyone's genome. CPGs are regions in the genome where you have a cytosine next to a guanine, and in these regions you might have methylation happening on the cytosine itself. And if you basically get the levels of methylation of these 353 CPGs, and you basically they created an elastic net model which can predict chronological age. So it was trained to predict chronological age to start with, and they saw that actually these models are pretty good at predicting this. And then from that, they took the next step and said, okay, if we find a difference between what the model is saying and the actual chronological age, what does that mean? And those are the measures that they understand as epigenetic age acceleration. So this clock, it has some features that are quite interesting. For example, it was developed using 50 different tissues and cell types. So potentially the model is supposed to work across different cell types and tissues. They've done studies where they've shown that this epigenetic age is actually heritable. So up to 40% of this acceleration or how fast you age compared to your chronological age can be traced back to your genetics in adults. It has been shown to be associated with different forms of health, including cardiovascular health, and it can be used to predict the incidence of different human diseases. And they've done a lot of studies, mostly in vitro, where they've shown that actually epigenetic age per se is not the same as telomere length. Even though they're correlated, they don't seem to represent the same biological processes. And the same for cellular senescence. They're actually different things. And in the same study from the Horvath group, they actually also showed that epigenetic aging is associated with mitochondrial activity and some form of loss of pluripotency by cells. So when we decided to start this study, we actually had two main hypotheses. The first one was that potentially plaque-specific epigenetic aging, so epigenetic aging measured in atherosclerotic plaque using DNA methylation would reflect disease progression beyond chronological age, so potentially could be a better predictor of risk and of disease itself than chronological age, given these features that I mentioned before about biological age. And then we had a second hypothesis, and this is because part of our lab dedicates a lot of efforts to understand sex differences in cardiovascular disease. And we hypothesized that given that there's a delay in atherosclerosis and the fact that it's been shown that sex hormones have a interrelation with epigenetics, we postulated that plaques in women would show delayed epigenetic aging compared to men, so they would age slower epigenetically than men's plaques. So basically in this study, the most of the data that I'm going to show you comes from the AtherExpress. So this is a large biobank of carotid endarterectomy patients. So these are patients that either have had a stroke or are at high risk of having a stroke or other complications from carotid stenosis caused by plaques. And these patients undergo a carotid endarterectomy where they basically have a surgery where they remove the atherosclerotic plaque. And then we have a protocol in place where we characterize these plaques starting from histopathology. So we take what we consider the culprit lesion, so the site of the plaque with the highest stenosis, and we use that for histopathology studies. And then we use contiguous sections of that same plaque to produce different sorts of multiomic data sets. So most of the data I'm going to present you today is on this biobank. And for this study, we actually had 337 male plaques in which we had measured DNA methylation and 148 female plaques. And these patient numbers actually reflect the distributions that you find in these populations with carotid with more male plaques presenting in our biobank. So we actually calculated epigenetic aging from these plaques, and we used the 450K human methylation array for this, which is the data that we had at the time. And then, as I mentioned, because we have a lot of information on these patients and on these plaques specifically, we actually could relate that epigenetic aging to all sorts of things, including clinical outcomes, risk factor profiles, characteristics of the plaques, but also transcriptomics and including single-cell RNA-seq. And just to give you first a summary of what this biological age looks like in our cohort. So this is the chronological age of our patients. You can see that they're quite similarly matched, the males and the females. Then we calculated this biological age. So this is the age predicted by the Horvath clock based on the plaque. So in this case, this is the biological age of the plaques. And you can already see that females indeed seem to be a bit younger predicted, even though their actual chronological age is very similar to that of males, and males tend to be predicted a bit older. And then here's just a spread of chronological age and biological age, and it doesn't seem to be something specific about a certain group. There seems to be quite good correlation in this case for both males and females. And then what we calculated is the epigenetic age acceleration. And in order to do this, what we constructed is a linear model where you basically construct a model using the biological age, in this case, the Horvath clock, predicted based on the chronological age as an exposure. And what you do is then you take the residuals of this model. So you subtract the biological age of an individual. Sorry, you subtract the predicted age within this model for that given individual from the biological age, and this is epigenetic age acceleration. So this is basically a measure of how much accelerated you are compared to your chronological age, but then subtracting the effect of age. We have to do this because from many different studies where you try to look at biological age, there's this phenomenon where people who are older are more likely to be predicted younger compared to people who are younger who are more likely to be predicted older. So by using this construction, we actually can remove that effect and really look at epigenetic age acceleration independent of chronological age itself. And when we look at this, we can see that actually males are predicted roughly almost on the spot. So they are predicted with an average of, I think it was 0.4 years. So basically they're on average half a year older than their chronological age. And for females, we actually saw that they're predicted, it was 1.8 in this case. So there was almost a 2-year difference between males and females. So females were predicted younger, 2 years younger than males based on their plaques. So this kind of pointed already that potentially female plaques are biologically younger than male plaques. So now that we know that you can calculate epigenetic age from the plaque, we wanted to know whether it had any relevance clinically. So does it matter whether you're epigenetically accelerated or decelerated compared to your chronological age? And for this, we constructed a Cox regression where we actually used a measure of composite event. So this includes MACE, so major adverse cardiovascular events, but it also includes major other appointments at the hospital, including major appointments for limb surgery, for example, and other serious vascular complications. And what we saw is that within this model, we found a positive correlation, a positive relationship between EAA and the log relative hazard ratio of these events. And this was linear, and we could see that basically the higher your EAA, the higher your hazard, but also the lower your EAA, the lower your relative hazard. So basically, it seemed like there was a quite strong relationship between how accelerated your plaque is and how likely you are to have a secondary event after the surgery. So in order to understand what properties or what risk factors might be associated with this epigenetic aging in the plaques, we actually basically dichotomized this variable. So we used a zero cutoff. So basically, if you're positive, if you're predicted older than your chronological age, we consider you to have epigenetic age acceleration. And if you're predicted younger than your chronological age, you're considered to be epigenetically decelerated in your plaques. And then when we split these, our patients based on their epigenetic age, we actually saw that patients, well, something that we had already seen, that being a male is associated with a higher epigenetic age acceleration. Same for BMI. So people who had epigenetic age acceleration had a higher BMI. and they had more likelihood to present with diabetes, so a higher prevalence of diabetes within the epigenetically aged groups. So that was already quite interesting. It seems like there's something about potentially risk factors that have to do with inflammation within these patients that seems to be associated with epigenetic age acceleration. So we then constructed a model will be adjusted for this, just to make sure that what we're seeing is not confounded by these risk factors, because some of these risk factors we know are associated with secondary events, such as sex, for example. And we saw that when you adjust this model for sex, BMI, and diabetes, we actually don't still find a significant association with having an event, well, a composite event in the 3 years after the surgery. So we thought, okay, this is very, very strong, but we still wanted to make sure that we could characterize this properly. So one thing we did is we had epigenetic age acceleration measured in blood as well for a subset of these patients. And although they were correlated, we actually found that plaque epigenetic acceleration was a much stronger predictor of secondary events than blood epigenetic aging was. So if you put them both in the same model, we actually saw a very strong effect for plaque EAA, but a not so strong effect for blood epigenetic aging. This was also independent of plaque composition. So we know within our cohort, we have very different plaque phenotypes, and we characterize those ranging from fibrous to atheromatous. So whether there's a lot of smooth muscle cells or a lot of lipids in these plaques. And we actually saw that this effect was independent. So whether you had a plaque epigenetic age acceleration, so having an epigenetic age acceleration was related with outcome independently of whether you had a fibrous or an atheromatous plaque. This was also independent of the presence of plaque hemorrhage. So this is a very strong predictor of secondary events in our cohort that has been studied widely in other publications and in other studies. And it has been replicated also using imaging techniques that can detect these hemorrhage in the plaques. And we saw that if you put these two things in a model, they're both significant with a very similar hazard ratio. So it's almost as bad for you if your plaque has hemorrhage as if your plaque has EAA. And if it has both, it's actually even worse. So this was quite interesting that we could pick up something that we basically couldn't see before, because this was one of the strongest predictors in our cohort. And we didn't find actually any associations with the hallmarks of plaque composition that a lot of people look into. So whether you had fat or plaque hemorrhage or collagen or calcifications or the amount of smooth muscle cells that you have or the amount of macrophage contents that you have in your plaque, they did not seem to be related to epigenetic age acceleration itself. So this was a bit puzzling for us, because normally we find quite strong associations with these factors, because these are normally the things that determine whether your plaque is high risk or not. So we decided to dive a bit deeper into the molecular mechanisms of this. So we first thought, okay, these CPGs which are distributed across the genome seem to be strongly predictive of outcome, but do they tell you something else about the biology that is associated with this epigenetic age? So what we did is we took this methylation data set and we subtracted these CPGs, which are predictive of chronological age, and we performed clustering based on with all the other CPGs in our data set. And what you can see here is that actually there's a clear clustering that basically separates males and females, and that is known because methylation patterns are quite different between males and females, and that has to do with, for example, the sex chromosome and the different levels of methylation related to the X chromosome inactivation. But actually, within the different clusters of males and females, we could see that, and especially for females, that the patterns in the DNA methylation actually quite well separated these plaques in accelerated and decelerated. And it was kind of also similar for males, where you found a relative degree of separation with some clustering appearing in some areas of the plot. So these told us that basically there's a lot of epigenetic status in the methylation of these plaques that seems to be also related to epigenetic age acceleration. So it's not only the CPGs, but it seems to be a genome-wide effect that you find. We actually looked at if you link these CPGs to genes, we wanted to see, okay, what processes do we find to be dysregulated? What processes do you find to be different between the plaques that are accelerated and decelerated? And what we found is that hypermethylation, CPGs that were hypermethylated were linked to genes that had to do with processes that you could consider related to smooth muscle cells. So you have a lot of extracellular matrix organization coming up, muscle cell development, muscle contraction, and so on. While the CPGs that we found hypomethylated in accelerated plaques actually had a lot to do with immune processes. So you can see here a lot of cytokine production, immune response, T cell activation, and so on. So it seems to have two ways. So it has a component of mesenchymal, like smooth muscle cells and other cells, and it has to be a component of inflammation as well. So next, within our cohort, we also have a quite large cohort of plaques where we've done bulk RNA-seq, and these are largely overlapping. So it was about 85% of the plaques for which we had methylation. We also had DNA, RNA-seq. And we did a differential gene expression between these two plaques, and we found a lot of genes to be highly highly expressed when the plaques were accelerated, and we didn't find so many that were actually negatively associated with acceleration. So all these genes that you find here, and we've highlighted some here, are genes that are more highly expressed in plaques that are epigenetically accelerated. We threw these into different enrichment databases and performed enrichment analysis, and we could see that there's processes that came up quite consistently, such as epithelial-to-mesenchymal transition, oxidative phosphorylation. We also found some evidence for TGF-beta signaling and TNF-alpha signaling. And these kind of hinted to us that potentially these processes might be associated with plaque epigenetic aging. Within this data set, as I mentioned, we also have data for single-cell RNA-seq, and this is from the same plaques for a subset of those, actually 46 patients. We managed to generate single-cell data, and we identified different clusters of cells within these, including immune cells and also mesenchymal cells. And what we did is we basically took these genes that I presented here that were highly expressed in accelerated plaques, and we projected those into the single cell. So basically you took all the genes and you calculate what they call a module score, which is, if you like a combination of the expression of these genes. It's not exactly an average, but it uses a methodology similar to principal components. So it will basically look at the principal component of the expression of all these genes across the different cell types. And what you can see here is that actually these genes seem to be mostly expressed in smooth muscle cells as well as endothelial cells within our data set. So that told us that potentially the processes that seem to be more activated in these accelerated plaques might be related to these two cell types. And here I just presented some of those genes which we found to be differentially expressed between accelerated and decelerated plaques, and you can see that they express across different clusters of the endothelial cells and the smooth muscle cells, actually some of them being quite specific for some of the subclusters within those cells. So yeah, I want to make here a little outbreak. Maybe if there's any questions, I'm happy to take questions. And then I'll continue a bit more on the study as we, and I'll explain a bit more about like the publication journey, if you like. So yeah, but first, if there's any questions.
If everyone agrees, I think we can keep those for the end.
Yeah.
Yeah,
Okay.
yeah, go.
So yeah, they actually asked me to sort of explain a bit of the publication journey for this paper and how for example, the review actually might have impacted the paper itself. So what I presented up until now is what we had as the first submitted article. So it was a combination of bulk RNA-seq, single cell, and the epigenetic aging, and a bit of the clinical relevance of it. So in this actually study started in late 2020. So Robin Harman, who was a postdoc here at the UMC, started the work. And then through a couple of years, we actually, in several work meetings dedicated specifically to this project, we worked together to push this project. And at the end of 2021, actually, Robin left for industry. So he went to work at a pharma company. And then the work was passed on to me with like a rough draft of the paper and but some work to be done before submission. So we changed a lot in the paper. So that took actually quite a long time, about a year and a half, before we actually had something that we were happy to, and we submitted to circulation in February of 2023. And we got the first decision letter in April of 2023. I had to dig up a bit of my emails, and there had been two reviewers. They came back with major comments, but they allowed us for de novo submission. So we had virtually time to work on it because they were going to consider it as a complete new submission. Yeah, and I basically got extracts from some of the comments that they got. So sometimes it's just the reviewer comments. Sometimes actually the editor takes the time to sort of summarize and decides, okay, you need to work on these, maybe not so much on these. Actually, in the editor comments, the main things that they picked up was in the first draft, we actually had quite a lot of emphasis on the fact that this was the epigenetic aging that we found in plaques was actually more specific and relevant than the one that we had in blood, but they had quite a lot of comments on that. You can see that most of the thing the editor actually mentioned was about the blood EAA, so this epigenetic aging measured in blood. And then there was also some comments about overselling some of the results and stuff, which they always do. But yeah, we actually decided to address this by basically cutting off a lot of the blood EAA. So we took away a lot of the emphasis on that and made some of the statements a bit less definitive on that. But then one interesting thing that happened is the reviewer too only had two comments, and I feel like they can be nice if they're good comments, but they can also be quite nasty. And I feel like this one actually, the reviewer too tried to shoot down the paper. And I think if you've seen a lot of reviews, sometimes they do that by either highlighting inconsistencies on the analysis or basically mentioning that there hasn't been a lot of work done to validate some of the findings. So that's what he wrote. So in the first one, he basically said that there was some discrepancy between the RNA-seq and the methylation, and we didn't really have an explanation for that. So we kind of did a bit of work to explain that. And then the second one was about whether we could find to have any gain of function or loss of function studies. So basically, if you mess up these cells that we had identified that seemed to be important for EA, whether you could find evidence that these cells actually change in their epigenetic aging. So that's when we actually brought in some of the lab validation that actually made it into the final paper. So we, at the time, were doing a study on EndoMT, where we also had already the whole setup done in terms of the cells, and we just decided to add, we were doing it only for transcriptomics, but we decided to add some DNA methylation to it and see if we could actually validate some of those EndoMT signatures that I presented before. And in this experimental setup, what we did is we took human coronary artery endothelial cells and we exposed them to either a control medium, 72 hours exposure of TNF alpha at 10 nanogram per microliter, a similar concentration of TGF beta, and then a combination of both. And we did RNA-seq and DNA methylation after 72 hours. And this model we've used quite a lot to look at EndoMT, because basically when you do this to the endothelial cells, you see them transitioning into a mesenchymal fibroblast-like state. But actually what we saw quite remarkably is that the cells that had been exposed to TGF-beta, whether in combination or independently from TNF-alpha, actually all of them showed an average increase of 4 years on the biological age compared to the controls. And that is only after 72 hours exposure. And you can see here that even though we only had 3 donors, the significance of the results was quite strong, especially because all of the donors actually had a positive trend within them. So this we incorporated into the paper, and I think it gave a really nice round story, especially around this endo-MT and validating the findings that we found in some of those RNA-seq expression analysis. So yeah, after we did this work, which was quite exciting because we didn't know what to expect, and it was at that point we felt like it maybe was a lost cause for the paper or for that submission, especially. With these positive results, we were quite happy that we would at least have a chance. So we submitted it again. So as I said before, the first decision letter was in April. In November of that year, we submitted it again where we addressed most of the things, and we actually excluded a lot of the blood things that I mentioned. We got a second decision letter from Circulation in December, so a month after we submitted the review, and they actually rejected the paper. So this reviewer too, who actually had been quite cutthroat in a way, he was happy with the paper, but they actually invited a new reviewer for the paper who had a lot of textual comments. We were actually quite surprised because I think it was one of these reviews where we could actually address everything pretty quickly, but they had a bit of nasty comments mentioning that our TGF-beta findings were predictable, even though I didn't agree at the time because no one had shown that you could that TGF-beta. They had shown that TGF-beta had a lot of epigenetic impact on cells, but that no one had shown that it drove in any way epigenetic aging. But they actually gave us a bit of the silver lining, was that they told us that if we addressed those comments, we would be able to resubmit to ATBB, and that would be considered our first round of comments for ATBB. We resubmitted in February 2024, so 2 months after. We got a decision letter in March with a minor revision, and they already invited the technical and statistical reviewers. And when they invite those people, normally it's because they're happy with it. They just want to finalize the final touches. And then in the 25th of March this year, actually, they accepted the paper for publication in ATBB. So I think that was about 4 years, or roughly around 4 years between the first idea about doing this and getting it published. So quite a long time, and people coming and going and so on. See, yeah, and then I have some conclusions. So basically in this study, we found that EAA was lower in female plaques compared to male plaques by approximately 2 years, but we don't know yet why that is the case. Patients with diabetes and a high body mass index had a higher plaque epigenetic aging. Epigenetic aging of plaque and not blood predicted secondary events in 3-year follow-up. And plaque EAA concurred with a high metabolism, epigenetic and transcriptional state in plaques, and is said to be underpinned by endo-MT specifically. And we validated some of these findings in coronary endothelial cells and using single-cell RNA-seq. And yeah, just finally, comments by the reviewers can be nasty, but I think sometimes they can be useful as well, because we probably wouldn't have done all that in vitro work if they hadn't asked us to do it, because at that point we didn't really know where to take it anymore. So yeah, and I think that's all I have. Yeah.
Well, thank you so much, Ernest, for your presentation, and I think everybody really appreciates the detail of the reviewers' comments, especially at the end, is something I think that we've all been through. So thank you so much for sharing with us your experience. Does anybody have any questions?
I have a couple. I don't want to take up. So what others think about it? Very, very interesting study. And let me start by saying that I'm very interested in NDT. So I gonna go that
Okay.
area. Um, so very interesting study throughout. And like, as you were saying, this is indeed novel. And earlier this year, another report came out, I think, again in the circulation family, describing how sex and NMT seem to be linked, as in a mouse model, they show that male mice seems to be prone to have a higher impact of NMT in the plaques compared to female counterparts. And I was wondering whether you guys, in the end, went in any way to try and assess directly the link between sex and NMT, because you did it through the epigenetic aging, but
Yeah.
I don't think you directly link those two things.
Yeah. So not as part of this study yet. So we do have actually an interest on NMT and definitely an interest on sex differences in atherosclerosis, because that's kind of what the lab of the PI I work with focuses on. But actually, so all of these studies that I've shown in the cells, at least, they were done in commercial endothelial cells that you buy from a
Mhm.
perspective. What we've done now is we have actually a protocol to isolate endothelial cells directly from these plaques. So when
Nice.
we get the plaque fresh, we isolate the cells, and we're preparing now a study where we're looking at endo-MT both in males and females and see whether we find differences. And that will include both transcriptomic differences, but also epigenetic differences. So yeah, definitely. We do have actually, I think it just came out as a preprint. We have a study where we've looked at basically it's kind of the same layers of data that we've looked at here. Actually, some more because we include some proteomics at the differences between male and female plaques specifically.
Mhm.
And in that paper, we find very strong links to endo-MT in female plaques specifically, actually.
Oh, okay. Wow.
So yeah, I know some of the mice data because I know the last one points to male plaques having more endo-MT, but we've had discussions with some collaborators in the Gary Owens lab where they had some male female mice and they had some really interesting data, especially around IL-1 beta in male mice and female
Yeah.
mice, and they seem to see actually the opposite, where IL-1 beta seemed to trigger an endo-MT like state in female plaques that was more prone in male than in male mice. But yeah, definitely we have an interest and we're working on it.
Yeah. Very interesting. Thanks for sharing that final bit as well. Yeah, because that paper that I was referring to actually left me a bit confused, because this difference didn't lead to a protective, atheroprotective effect. You know what I mean?
Yeah,
There was less NMT, but they didn't meant less athero. So I was like,
yeah.
I was not really sure where they're coming from. And I think I had another comment. Yeah, you mentioned as well, you guys did a great deal of work around like epigenetic aging, and mitochondrial activity, and loss of pluripotency as well. Can you like comment in any way around that? Because
Yeah,
that was
so
very brief but interesting. So
yeah, so that study is not from us. Actually, was led by Ken Raj and Steve Horvath from the Altos Labs, and
uh-huh.
there they actually do take they had a whole set of experiments where they looked at what things actually changed if you take cells and sort of age them in vitro. And what they saw is that if you, for example, they did a lot of work in keratinocytes where you take them out of the tissue and you put them in vitro and you can see them epigenetically age. And what they saw is that when they reach an epigenetic age of about 80, 90 years old, that's when they stop growing in vitro.
Mhm.
So there's something intrinsic to whatever processes they find that seem to be related to this epigenetic age. And that's where they found that some of the main things that changed in those cells were uh, related to mitochondrial activity. And
Okay,
also
okay.
the loss of stem cell pluripotency. I think that was because they also did measure the epigenetic age as they sort of uh, uh, what do you call it? Like when you get IBS cells and
Differentiate
you
them. Yeah.
push them. Yeah, exactly.
Oh.
Once you differentiate them, the further they are in their differentiation stage, the higher their epigenetic age. So basically, when they're committed, they saw that their epigenetic age was much higher than that in the
Right.
process of committing to that fate. So I think it was along that, but it's a paper in Nature Aging from I think the last author is Ken Rush, but it's a very interesting paper.
Thanks for
Highly
sharing.
recommend reading it.
Chris, I think I saw a hand raised from
Yes, we
Stefan.
have. Yes,
Yeah.
Stefan, go
Yes,
ahead.
hello. Yes.
Yeah.
Yes, that's me. Yes, that's correct. I congratulations on the story. Interesting. I have two questions. One concerning the blood. Did you see the blood did you study blood epigenetic clock, and is it also accelerated? You mentioned that some of the blood data was removed from the paper, but apparently you have some insight into this.
Yeah.
So I would be happy to hear the comment.
Yeah, so we did find so they are related, as in we found a correlation between epigenetic aging measured in the blood and epigenetic aging measured in the plaques. But the correlation was not as strong as you would expect. I think it was a 0.4, something like that. So I think it's you could say that it's probably very likely that if you have one, you probably have the other. Which one precludes which one, I wouldn't know. But the thing is that the relationship with clinical outcome, that was very specific to plaque epigenetic aging. The only thing is that the reviewers weren't very happy about the fact that we had almost 500 plaques for which we had epigenetic aging, but for blood we had many less, so we had about 100. And that was purely because, so at the moment you go into the surgery, they ask you for consent for plaque, but then for blood you have to sign a different consent. So a lot of patients didn't actually agree to give blood for research, or they didn't need to. So then you're not allowed to just take it for the study. But
so maybe the correlation was weak because of the lesser number of samples. So if you have really 100 and 100, did you did analysis 100 blood and 100?
Yeah, we did. So in that case, when you look at the comparison, the correlation was not great, as I said, and then the association with outcome was mostly driven by the plaque. You didn't find that correlation with the blood. So that's what we thought was quite interesting. But the thing is that I also think that one of the reasons why they did is because there's been a lot of really large studies where they've looked at epigenetic aging in blood, and they found a correlation with different, not specifically with the outcomes we looked at, and not in the population we looked at, but if you look at, I think it was in primary endpoints, they found strong associations with, for example, I think the incidence of myocardial infarction in other studies, I think I want to say in the ARIC, but I can't remember now. But yeah, so I think they were a bit worried that we were saying, oh yeah, blood is not important, it's only plaque, while there's studies that show that in many other cases, blood is actually important. That's why I think we had to tame down our statements. It's not completely gone from the paper, but yeah.
Yeah, and another question relates to the Horvath. So you use Horvath clock?
Yeah.
And you did wild DNA methylation studies. So I didn't get it. So it was how did you do it? What technically how you did analyze DNA methylation? Was it a matrix or it was the whole sequencing?
No, we did the chip array for 4050K array from Illumina. So there already you have.
It was a chip.
Yeah, it's a chip.
For how many places?
So so the chip had 450,000.
450,000. Okay.
Yeah. And then, but the clock itself only uses 353.
Did you try other clocks? Did you try other?
We did calculate other clocks, but they had a very strong correlation, and we decided to focus on one to keep the story simple. But we did calculate others, and they showed pretty good correlations with it. There wasn't any strong differences. Yeah.
Oh yeah, I see. So it was a methylation epic, infinitum methylation epic.
Yeah, chip. That's the one there. It's actually not the latest. The one they use now everywhere is the epic, which is that plus another 400,000. But that's the one we measured back in the day when they generated this data.
So it's so basically you have a lot of you said 30,000 and 300 or 400 are used. So you have a lot of not analyzed methylation site.
Not for the calculation of the age, but then we looked at the methylation sites on the rest of the genome based on whether you are aged or not on the clock. We look at what other things in the methylation was different, and that's where we saw a lot of signals from smooth muscle cells related processes and
All right, I
immune
understand.
processes as well. Yeah.
Okay, thank you very much. Congratulations.
All right.
Very
Thank you.
interesting work.
Thanks.
Okay, I see that there's a question in the chat from Ziba. Is it really? No, it is really an interesting study. I wonder if the high levels of TNF alpha or TGF beta in blood can predict the epigenetic age acceleration.
Yeah, that's a very good question. So we've actually done now some studies where we've looked at the plasma of some of these patients, and actually inflammation is definitely higher in those patients in the plasma, but we don't necessarily find the same cues actually. So we don't find TNF alpha and we don't find TGF beta per se. But we do have some evidence from so we also have proteomics that we've done in these same plaques, and there you find it. So I get the feeling that part of that signaling might come from the plaque, and part of it might come from the circulation. And in the circulation, we find more other types of inflammation. It's still inflammation, but it's more like IL-6 inflammation, IL-18 inflammation, but not so much TGF beta. So we think that might come from some of maybe from the vascular inflammation. So the processes that are happening within the plaque. Yeah, it's a very good question.
Okay, and we have another one here in the chat. Thank you for your presentation from Mohammed. I would like to ask a question. Would it be a good idea to explore the association of LDL modifications in plaques with EAA and determine the most aggressive LDL modification?
Yeah, so we did have, I mean, I'm not an expert at all in LDL, in lipidology. So I don't know exactly what you mean by LDL modifications, but we did have some, like the classical levels of lipids, like LDL, HDL, triglycerides, these sort of things. And they didn't seem to be strongly correlated with it. I know there's very strong links, obviously, with levels of LDL and inflammation itself, but in our study, that at least didn't come through. Although the link between inflammation and epigenetic aging was very clear. But maybe if we actually had access to some of the more specific LDLs, because when you talk to the lipidologists, they have all these different forms and versions of it that you can measure. Maybe some of them are actually related to this, but in our study, it didn't come through with the basic ones. That's a good question.
Okay, thank you. And I see Fabio has raised his hand. Fabio, go ahead with your question. No.
We cannot hear you if you are asking the question, Fabio. Maybe. Can I ask the question while Fabio maybe works on his connection? Thank you so much, Ernest, for this tremendous work and amazing data. I maybe missed something or misunderstood, but it seemed to me that when you did pure analysis on the epigenetics from plaques, it seemed that more story was related to T cells and T cells biology. And then when you cross over it with the single cell sequencing and transcriptomics, it didn't seem that the T cells, you know, the violent plots that you showed, the T cells were relatively, I mean, there was no changes. It's more like endothelial cells and smooth muscle cells. So if you can just maybe go again through this part of the story, just explain to me where these different, how you obtain something from the epigenetic point of view, and then later on the transcriptomics show different emphasis on different cells. Thank you.
Yeah, so normally, I mean, I think the key there is In the analysis we did on the epigenetics, so we actually had to a bit of push and shove with the reviewers on this. So in the epigenetics, you basically look at the methylation on a given side of the genome, and then you have all these annotations, which some of them are based on literature, some of them are based on actually a closeness to a gene, and that kind of determines whether a CPG is linked to a gene. So we basically use that we were very worried about making big statements about, yeah, this CPG is linked to this gene, therefore this is involved. So we actually, in that analysis, we consider anything that would come up there as potentially involved, but we didn't put a lot of emphasis on the directionality. But then when we looked at the, as you say, when we looked at the expression, then it seemed like another signal was coming through, and that could be for different reasons. So actually, I do think that both of those things are involved, so both the mesenchymal part and the more immune-related part. But for some reason, in some of the data sets, one of the signal comes even more strong than in another. And I know, for example, that if you look at things like proteomics in the plaque itself, there we find a lot of the things that make sense with the RNA, so things correlate quite strongly. But then if you look at the for example, at the plasma circulation, there you find a lot of the immune stuff. So I think there's some crosstalk, and the epigenetics itself has a bit of both, because the epigenetics will also be strongly determined by the composition of the plaque. So if there's more of one cell type, you'll find that in the epigenetics. While I think the expression is a bit more, yeah, maybe less in our expression, I have to say, because this is very specific to the plaques. When you work with these plaques, the tissue is so advanced, the atherosclerosis, that the tissue sometimes is very degraded. So for example, the quality of the RNAs that we get sometimes are not the best. So it could be that there's some biases in some of the different in some of the different compartments, so the RNA, the proteomics, the epigenetics, the signals might be different. But I think now that we've done, I haven't included there because it wasn't part of the study, but we've done all these other analyses using proteomic approaches, and the same signals keep coming up really strongly, but then you have some signals in some of the compartments, some in others. So I think you do need the whole picture to really make sense of what's going on. Yeah.
This is why I'm asking, because we currently did my postdoc at Luxembourg is focused on using epitranscriptomics, using nanopore to do the RNA sequencing and see how M6H changed in the correlation. And we use the PBMCs as a kind of classical approach. We didn't have plaques, nothing like that. But when we see the differential expression between people with myocardial infarction and stable coronary artery disease, we really see, again, one of the most enriched pathways is also related to TGF-beta. This is why it means, to me, it's a little bit interesting to go deeper.
So I have to say, like, the TGF-beta, apart from the in vitro work that I think for me really, like, I was not necessarily expecting that it was going to be such a clear and strong signal. We've actually done now some single-cell RNA-seq where we've selected the plaques based on whether they're accelerated or decelerated. And then the strongest signal by far is the TGF-beta, like the endothelial cells. In all of the 3 patients that we chose as being accelerated, you have almost like a specific cluster of endothelial cells, which is enriched for TGF-beta, that you don't find in the others. So I think there's definitely a very strong signal there with TGF-beta specifically. Yeah. So again, it's depending on where you look at but the signals seem to be really strong. Yeah.
Thank you. Fabio, can you join us? Try to.
Yeah, I'll try again. I don't know if you can hear me now.
Hear you now.
We can hear you.
Oh, okay, good. So, Ernest, congratulations, really beautiful work. I have no, probably didn't get very well beginning of your presentation. So I was puzzled because you show that epigenetic age acceleration in males is higher than in females, and that is not surprising. But then when you look at the value for females, it was actually negative, meaning the plaques were younger than predicted by chronological age. Isn't this surprising somehow? I mean, the plaque are younger than the rest of the organ, or does this mean that the clock works, but is somehow out of phase a little bit, and maybe there are better clocks for the plaque?
Yeah, so one thing that they've already showed in some of the early studies on epigenetic aging, so all the studies from Steve Horvath and so on, is that some of the clocks actually they predict females to be younger, and they couldn't really pinpoint whether that is a true effect or whether that is potentially some bias in the way the clock is trained. Because I think, from what I understand, all the clocks are basically predicted using both males and female tissues and cells and data. I don't think people have created sex-specific clocks. So we in the paper we actually took a really sort of politically correct stance on it. So we didn't say that that was a thing, or because it could be that it's just the clocks that are, because we didn't develop the clock itself. One thing that would be very interesting to do, for example, would be to look at whether, if you train a clock in females only and a clock in males only, do you still find those differences? So it could be that there's indeed something intrinsically younger about female plaques. I know also the composition of the cell types can affect quite strongly the clock calculations, and we know from a lot of our studies that female plaques are much more fibrous, so they tend to have much higher content of smooth muscle cells and collagen. So some of those things might impact somehow the calculation, because at the end is based on the biology that you have in those plaques. So yeah, I wouldn't be able to tell you whether. I don't think it was necessarily surprising, because they had reported it to some extent in other studies, but the amount of how different they are to us was quite surprising. But for example, one thing that we were trying to answer with this study is, a lot of people tell you, well, if you study females because they have 10 years before, you're just looking at the same plaques you're looking in males, just 10 years younger, because the plaques develop later in time. But what we see is not necessarily that. Like, they're not 10 years younger, and they're not, so they have differences, but they're not necessarily because they're younger plaques. So I think that's one of the things we wanted to try and address with this. Yeah, so I don't know if that answers your question. Sorry, I diverted a bit.
Yeah, no, very well. Thank you.
Perfect. Thank you. Let's end our session here. If anyone, I'm sure Ernest is more than happy to. If you have any further questions, if you can contact him directly. Thank you so much, Ernest, for taking the time to present this amazing work with us. Super interesting. Thank you very much. And thank you to everybody who participated and for the questions and the lovely discussion. I think this was quite a good discussion that we had today, and I'm super happy to see that more people are being involved in asking questions. So that's super fantastic. Fernando, I guess you have
yeah, on that note,
yes.
Some of you maybe already joined us in July for like the informal journal club chats format. And just please be aware that next month, on the 18th of November, we are doing another informal session, and we will be talking again about how to navigate PhD life. We'll let you know more about like which aspects of this we are talking about. But happy you guys enjoy the meeting, and hopefully see you all next month.
Can I just send these links to PhD students in our institution? It's open.
Yes, please.
The more the merrier.
Please do.
Please do.
I like this format, and I think it's an excellent idea. So I will be happy to transmit this info to the people.
Really appreciate that.
We are trying to engage more and more people that are younger, so it would be fantastic if you can spread the word around. Thank you.
This really is designed to help younger people.
And the idea is to present a good paper for the young investigation. Yes, it's exactly. A paper. Okay.
Yeah, so here
Exactly.
basically we
If-
have the first author usually of that paper, which is a young, is an ECR itself or himself or herself, like a PhD student or a postdoc like today. And yeah, so having someone young presenting an ECR is good for other ECRs to ask questions potentially. So the more ECRs are attending, and the more the questions that are coming, hopefully.
Fantastic.
Please leave your emails. For those of you that haven't, Melody made a comment in the chat section. Please leave your emails in the chat. That way we can add you to our mailing list, so you can have easier updates for the next upcoming chats, and also for the future journal clubs that we'll have.
I think I am already, but I will write.
Okay, perfect.
But I will write it in a
Perfect.
yes, thank you.
Great.
Thank
Have
you
a
very
great
much.
week, you all. Thank you for
Yeah.
joining
Bye.
today.
Thank you.
Bye, everybody.
Thank you all.
See you next time.
Bye-bye.